Lab on a Chip Systems and founded BiFlow Systems in the year 2011, I believe. Thanks, April, for starting the recording. Just to state as well that we will be recording this webinar. So yeah, and it will be available for you to access after this live webinar. And with that, I would like to give the floor to Jörg Nestler for his presentation.
Thank you, Divish, and welcome everybody for this webinar, the first one in a series of webinars, as Divish just explained. And of course, it's also interesting and nice and some kind of honor to be the first one having the opportunity to present some topics. Today's webinar is on Lab on a Chip system for molecular diagnostics, and after a very short, very brief introduction on what BiFlow systems is, I will go then to Lab on a Chip systems in general. I don't think that I need to explain here for that audience, as you joined that webinar for some reason, what a lab on a chip system is. And then I will focus a little bit on topics like integration, multiplexing, and speed, and then end with two examples. One is from another project with some other partners, and then the respiratory example is from the next-gen microfluidics project. So BiFlow Systems, I think it was said already, was founded in 2011. We are currently 18 employees and have a manufacturing space of around 250 square meters. My contact details you can find here in case you have any further questions, which we cannot answer today for some reason. What are we doing? We mainly focus on microfluidic cartridges, as you can see on the left side. So they all look a little bit like what you can see here. They all have the same working principle behind. I will explain a little bit later. We also have our own instrumentation. The old instrument I didn't show on that slide. This is the new one, which is coming soon in the next couple of months. And you see some results you get from this one. We, of course, provide custom solutions for our customers to research and development for them. And of course, we have fabrication capabilities, automation of the fabrication, but also some handmade fabrication steps in-house. So that's all on BiFlow systems. And what we are actually doing, you will see in the next couple of slides. Before I start going into detail, just one slide on Lab on a Chip systems to make sure that we understand the same thing when we talk about that. So in general, these are small microfluidic systems that can perform one or more laboratory functions. So in theory, they should be able to start with a sample and just give you the result. What is important here to understand that lab on a chip is a little bit misleading, as typically this is not a chip, but a much larger cartridge made typically of polymer, and it's not like a millimeters wide chip, but it's more in the centimeters range, the height maybe in the millimeter range. And today, of course, we will focus on a special application for such lab on a chip systems, which is molecular diagnostics, meaning nucleic acid based. And I will not focus at all on lateral flow strips, which you may also consider by the definition above as a lab on a chip system, but that's definitely something we will not talk about today. Okay, now, if it comes to lab on a chip systems, for whatever kind of application, there is always the question what to integrate in such a lab on a chip system. And of course, it always helps if we look into what is done in the laboratory. And normally you have steps like the lysis of the sample. You have a purification step for your amplification, and depending on whether the detection is part of the amplification or not, for example, in case you have to hybridize your amplicons to a rigid surface, then there's also the detection hybridization step. On the top, you find some activities which need to be performed, which include pumping. I mean, in the laboratory, this could mean pipetting, valving, which can either opening a bag or a bottle or whatever in the laboratory, or close it. or seal it, and you need heating steps for some of the different steps. And looking at this, you may ask then, okay, for lab on a chip systems, what of those functions I really want to integrate into the lab on a chip and what I would like to keep outside? For example, in the instrument which runs that lab on a chip and performs whatever is going to be performed on that lab on a chip. So that's an answer which always needs to be asked in the beginning. And there are some advantages and disadvantages of integrating everything or leaving out everything or just part of it. Of course, the less you integrate, the cheaper the disposable, so the lab on a chip as such, becomes. However, this puts more complexity to the instrument, meaning typically also that there is more maintenance required, and also that the instrument typically becomes a little bit more heavy or bigger. On the other hand, high level of integration poses also some advantages. Of course, it's very simple to control the disposable because the only thing ideally you need to do is, for example, to contact some electrodes on that disposable. And the rest is done internally into the disposable. Of course, the instrument itself can also become much simpler. You can even think of controlling such a cartridge then by your mobile phone because you don't need to have that many additional components around it to run it. And of course, there's then also less maintenance of the instrument, but the drawback is the disposable may become a little bit more costly and a little bit more complex. Now, I will show you our approach towards full integration because we believe that the future is still a highly integrated, a full integrated lab on a chip system because there is the tendency, even legally and regulation-wise, this is still a difficult topic, but you can imagine that in, I don't know, 10 years, maybe everybody has its own self-test capability, which he can run by mobile phone. So it's some kind of democratizing diagnostics, I would call it. So what we definitely don't want to see with integration and the lab on a chip system is that kind of tubing you see on the left side. We don't want to see external pumping, but really want to have everything integrated. And the way we do that is that we have here on the top an electrode substrate where some electrodes are covered by a hydrogel. And then there's a separation membrane, and on top there is a reservoir which can contain liquids. What is worth to mention here as well is this green membrane, which is forming a normally closed valve. And then we have sensor region or other reservoirs, which we can heat by heaters, which are also integral part of that whole system. And in case we use optical sensors, we may have here a sensor foil, which forms the sensor with the microarray, but I will talk about that later. Now, if we operate it, meaning that we apply a certain electrical voltage here to that hydrogel, we generate a gas which deflects that membrane into the reservoir, and that pushes the liquid out. I mentioned that there is actually a valve here, normally closed valve. So this is actually not opened by the pressure, but opened by some other method in the instrument, so that it frees the path from that reservoir to the channel system. And then the liquid can go, for example, to the sensor area or some reaction areas. And the whole thing is basically controlled from the backside by electrodes, which are contacted in the readout and control instrument. Here you can see it in action. I hope this can be seen. So here's the sensor area, and you see that the red liquid came here, and there's a magic which basically makes the color disappearing in this area. And you see how it slowly gets transparent, which is due to the temperature of the heater underneath that area. So the grayish part is actually the heating area. So what you actually see here is the pumping itself with this thermochromic liquids and the bleaching of that thermochromic liquid due to the certain temperature which is applied here to the liquid. And here, by the way, you can also, starting from the beginning, you can see the liquid moving in the waste channel. Now, I was talking about also on reagent storage, because for whatever kind of assay you need to use some reagents, you can either use them as liquid reagents, so basically stored in the liquid reservoirs we have on the cartridge, but you can also think of dry reagents. And the way we are doing this, we have a porous matrix here, which is filled with the dry reagent. Then we open the valve and actually rinse the liquid through that porous matrix, and by that release the material. Now here you can see the two yellowish and the two bluish dots. And this is the porous material containing, in this case, some yellow and some blue color. And then we rinse that reservoir here where the green arrow is showing. And then you see here how actually the sensor area gets yellowish. And on the left side, you see again that this former yellow spot now became white, because all the yellow color was actually rinsed out by this buffer in the reservoir. And in a few seconds, you will see the same thing with one of these blue dots here, this blue porous membrane. I think in 2 or 3 seconds it will start. And then please look to the right side. Now it's coming to the right side, how the color changes in the sensor channel. So this is the way you, besides liquid reagents, you could also store dry reagents in that cartridges. Speaking of integration, despite of reagents and heating and valving, of course, you also need whatever kind of biosensor. We typically use with fluorescence microarrays, but of course, the technology is also compatible and suitable to run other biosensors, like other photonic biosensors or electrochemical biosensors. And especially electrochemical biosensors in combination with the cartridge technology, which requires only electrical interface, has the advantage that this vision of running such tests purely by your mobile phone becomes more true in the future. Now, I was mentioning multiplexing before, and I think that's another topic. Despite of the level of integration, where I was saying, okay, we envision a high level of integration, multiplexing is something to consider as well. Because you can imagine there are tests which just go for one parameter, but you can imagine tests which are looking for much more parameter. Now, speaking of multiplexing, if you look a little bit to the landscape, here in this diagram, you see on the x-axis the speed of the test, and on the y-axis the numbers of parameters. Now, if we look to the commercial landscape, you will find many commercial players, especially pre-COVID pandemia, more on the left side. So there are a certain number of players who are able to measure more than 5-6 parameters at the same time, just by the technology they're using. However, their tests are typically quite slow, and slow means slower than half an hour, typically 1 hour or more. In COVID pandemia, actually, there was a demand of molecular diagnostic testing for SARS-CoV-2. And there you don't need a lot of parameters. What you need is speed and, of course, a more lab-like quality of the test, as usual. And there a lot of technologies emerged from, I would say, an early phase quickly to a more major phase. But those tests are typically for a low number of parameters, but they are focusing on speed. And the way this is typically done is by some isothermal amplification method here, which can be quantitative by, for example, some fluorochrome, like in quantitative PCR. However, what is still quite empty is the combination of having a high number of parameters, so high level of multiplexing, and a fast test. Fast test means 30 minutes or quicker. And that's definitely something which is highly interesting, because there are plenty of applications which require a larger number of parameters, and which can simply not be addressed easily by having either a slow test or a fast test, which just can go for a couple of parameters. Looking a little bit differently into multiplexing, I will here, I compare here the Cube PCR, which of course can run yeah, if you can parallelize it on a microtiter plate, it can run several samples a day, so you can have easily more than 100. But you are kind of limited by the number of targets per run, because normally you're limited with the fluorophores you're using, and of course also all these effects having too many primers in your sample or in your assay. So you are limited in the typical PCR with the number of parameters you can go for. On the other hand, qPCR is suitable for having a portable device, so this is quite interesting then. But as I said, it's limited with the number of parameters to go for. Now, these highly multiplex molecular point of need diagnostics can actually, and I will show you later how we can do that, can actually have much more targets per run. And of course, you can also have more than 10 to 20 samples per day in one instrument, and you have a high level of portability. So this is mainly to show how also NGS, next generation sequencing, differs from what we are doing and what PCR is doing. And of course, there are a lot of people seeing the bright future for NGS, and there is definitely a bright future, and there's a lot of miniaturization and simplifying of use there. But there's still some kind of gap in between what is done in the laboratory with respect to PCR and NGS. And this gap can be, for example, covered by such a highly multiplex molecular point of need test. Regarding multiplexing, there are a couple of applications, I mentioned that already, which require a large number of parameters to be identified. And one of these applications is the identification and differentiation of species. Could be bacterial species, fungi, virus types or their variants, or different animals or plant species. And another application is the drug resistance testing, so identifying drug resistant genes. But sometimes it's not only the gene, but it's required to detect certain point mutations. So this is not easily doable by one or two parameter test. Now, what ways of multiplexing do we have? The one I mentioned already is to have a multiplex qPCR or some other quantitative isothermal amplification, basically a single well which contains different primers to amplify different regions of the genome. But this is, as I said, typically limited to, yeah, I would say around about 6-plex, which is already quite challenging. So you hardly find more than 3-plex. And actually, this option is also very limited for most of the isothermal methods, so it cannot be easily used for most of the isothermal amplification methods. Another option to go for is to have either a single-plex qPCR or an endpoint PCR, or isothermal amplification, but multiple wells. So it's easy to just spatially separate the different reactions into multiple wells. There's one drawback of that. I mean, in a lab, in a microtiter plate, it's easily possible, but one drawback is in microfluidics in a small cartridge. This separation into different wells is usually a quite challenging task. It's doable and there are approaches to do so, but it makes the whole thing a little bit more complicated, so it's not always the best way to go for. So this is all the multiplexing during amplification. What you also could do is, of course, you can have multiplexing during the detection. For example, by having a microarray of probes which are specific to a certain part of the amplicon. And this is done typically by fluorescence microarrays, but also by most of the more array-like electrochemical biosensors. And besides that, multiplexing during amplification and multiplexing during detection, of course, you can also have a combination of both. And this is what I will show you here now. It's a little bit messy on that slide. So I would explain a little bit the headline here. So this is actually the black headlines are more the lab scale procedures, while the whitish ones are more point of need, or are point of need. And here you have endpoint PCR followed by a microarray. So you can do this in the lab, but this typically, the PCR itself takes some while. It's of course, due to the microdata plate, easy to parallelize. As it's a traditional PCR, you can also have large amplicons, more than 2,000 base pairs, capable of multiplexing. It is as well. I mean, especially if you combine it with a microarray. And of course, you can look for a large number of targets as well, because you can use the multiplexing from the PCR and combine it with a microarray. But that's the lab scale. qPCR is slightly different, especially in the sense that the numbers of targets are limited, at least if you run it in just one well. And speed may be a little bit quicker, because you more quickly see that you amplified something. I will mention another type of PCR, which we call a pseudo isothermal qPCR or PCR. You will find that later in the slides as Psi PCR. This is actually kind of a stationary heating, but still you have running the liquid through different temperature zones. So by that, you can avoid the actual cycling of the instrument, but still can run a classical PCR. And that method is slightly faster than conventional PCR, so around 2 times faster, but is more or less compatible with a classical PCR. So it's easy to transfer from classical PCR to this Psi PCR. And now this Psi PCR can be used together with a microarray, for example. But of course, despite of that, you can also run a classical isothermal amplification or a point of need isothermal amplification with a microarray. So both is possible, either a Q, also quantitative isothermal amplification, or an isothermal amplification with a microarray. Now, the major message of this slide is all of the methods have drawbacks. Depending on your application, they are of course very useful. But if you want to have something very versatile, which unfortunately is not on the market yet, you can combine a very quick PCR, like I mentioned before, with a microarray, and you come up with a high level of multiplexing. You can still go for a large number of base pairs, so large amplicons if this is required. You can easily do multiplexing. As it is point of need, it's easy of use, and it's still quick. And that's the important thing here. Looking into what is typically used for what, of course, I mentioned already, most of the SARS-CoV-2 lab-based tests were done with qPCR, while the majority of the point of need molecular diagnostic solutions for SARS-CoV-2 was made by isothermal methods. And basically, we focus more on the rest here. So speaking about speed, I will give you an example despite of that special PCR method, which is also important and which contributes to the total time of the assay, and where, however, microfluidic has some advantages. Before I come to that, I will introduce SNP detection. I mentioned already that SNP detection, so single nucleotide polymorphism, is important for some applications to distinguish, for example, between several species of animals or bacteria which are actually close to each other but differ in some mutations. And this is done by spotting on a sensor surface the same oligonucleotide, which differs only at one position, the SNP position. And just if you have a perfect match, like here the right one, the T1, you get a strong signal, while the others are much weaker. So you will have some kind of binding, but it's not perfect. So you wash away also a lot of fluorophore and oligos here. And by that, you can compare all those results. So it's not quantitative, but it's a relative comparison between those 4 spots in that case, and you get the perfect match. This is important to understand the next slide. Because there we have a microarray which was focusing on species and antibiotic resistance detection. And in the laboratory, this assay typically took 120 minutes. Now we transferred that to a cartridge, and we started with more or less what we assumed in the lab would be the time for, or not assumed, actually this was the standard in the lab to do hybridization on a microarray of 60 minutes. Now we were able to get this down on the cartridge to 15 minutes, finally even to 5 minutes, but we left some safety margin here, so 15 minutes gives at least still nice results. And what you see here are those SNPs. So this is important because if you lose specificity here, then actually you cannot easily distinguish this perfect match. So this is a problem if you go down to 5 minutes or less. So actually those bars are looking more closely to each other. But with 15 minutes, you can easily say that you can see that you can distinguish between those different probes very, very nicely. So a reduction by a factor of 4 to what we started with, and actually including all the handling time and washing steps and so on, it was finally even a factor of 8 compared to the laboratory. Some other aspects of speed I will show you when I talk about some applications, and I will start with the identification of bacterial species and antimicrobial resistance for a specific use case, which is diabetic foot ulcer. Diabetes actually is a big issue, a big burden to society. In a lot of countries, or areas of the world like US and Europe, of course, but even worse in countries like India, for example, which is one of the reasons we collaborate on that topic with Indian companies and institutes. And actually, the problem with diabetes is, especially type 2 diabetes, that it can lead to a loss of sensation in food, in the food, for example, and this finally can lead to some ulceration. And after that started, actually amputation will be typically the result. So this is really a big issue, and you would claim that this is mainly an issue in countries with not as good healthcare system like in the Western countries, but also in Germany. And I've heard from a person from my team actually that she is aware of such a case. So this is really even in Europe and Germany a big issue. And of course, those are infected wounds, and infected wounds can be infected by different types of bacteria. And they can also be resistant to antibiotics, and then definitely amputation is the only choice at the end. Speaking of antimicrobial resistance, it is believed that if everything continues like it is now, actually the dominating disease which leads to death in 2050 will not be cancer anymore. Or other diseases, but actually deaths due to antimicrobial resistance, because as you know, drug resistance increases. We still lack of new novel antibiotics, and this may become the end of the antibiotic era. I hope this will not happen. However, we have something to do here, actually a lot to do here. What you see here is the so-called escape species. So these are the species of concern with respect to antimicrobial resistance. And of course, we are going to look for that, but only the ones which are present, of course, in the diabetic foot ulcers. Now, what you can see here is how the workflow looks like. You start with a wound swab. You do a lysis, and the lysed sample then undergoes, in our case now, an isothermal amplification to these other amplification methods, this Psi amplification I come to later. And this is then hybridized and detected. And this is actually the instrument of our partner at Shira Labs, the first microfluidics company of India, round about our age. So they have an instrument running our cartridges here. And this is basically done. This is a colleague from Microarrays Labs performing that assay. And we were able to successfully amplify on the cartridge in the reader and hybridize, got the microarray readout from the reader, and of course successfully then also detected the different pathogens and resistances. To give you an example, here you see Acinetobacter baumannii with one very famous resistance, which is New Delhi metallobeta-lactamase. And on the right side, you see a Staphylococcus, in this case, Staphylococcus hominis, which is methicillin resistant. And this is what you get from the instrument, from the software. So you have, of course, the spotting pattern, you have some pattern recognition, you have a quantification, and then there is a decision tree afterwards telling you what you actually found in your sample. We did this a little bit more extensively with different types of resistances, like or resistance genes like MAC-A, WIM, OXA, NDM I just showed you, and also different types of bacteria species. So this is one example, which are bacterial species, but of course this also works for viruses. But there we used a slightly different approach. And before I go into a little bit more detail, I will focus on some aspects of that hybridization on the microarray. Normally, when we do PCR or whatever kind of amplification, we get double-stranded DNA out of the PCR and use that for hybridization. This works, but signal intensities are typically quite low because the second strand also always competes with the oligo, the probe on the sensor surface. So it would be much better to have single-stranded DNA. And of course, you can either denaturate before hybridization, but this involves a high temperature step on the cartridge, which, if not necessary, could be avoided. Or you can use an amplification method which generates a single-stranded DNA. And one method to do that is the so-called late PCR, the linear after the exponential PCR. And this is kind of an asymmetric PCR with a special primer modification. And so actually what you get is you have this starting curve from a symmetric PCR, so exponential phase, but then it goes into linear phase, which generates single-stranded DNA. And even though not all of your amplified DNA is single-stranded, you have sufficient amount of single-stranded DNA to give you a beautiful and nice hybridization result. And the difference you can see here, so this is still classical late PCR, so classical PCR, but with late primers on the left side, and on the right side, you see a typical standard symmetric PCR. And what you see here are different viral loads of SARS-CoV-2 in the sample. And you nicely see that with late PCR, after hybridization on a microarray, you get much stronger signals than with a symmetric PCR. Now, the same late PCR can be done with this Psi-PCR. And to explain a little bit the potential of that, on the left side here, on the left picture, you see a standard thermocycler, while such a Psi-PCR thermocycler can be much, much smaller. You have much less heating elements or smaller heating elements. You can even power this by battery. And what you see here on the right side is a CubePsi PCR. So this is a quantitative one, which gives you curves which look more or less similar to what you're used to from a classical PCR, classical qPCR, with the difference that it is slightly faster. And the main advantage of that is that it's much easier to integrate into microfluidic cartridge compared to cycling between temperature zones in a classical PCR. So it's more comparable to an isothermal PCR, with the difference that you can easily transfer your lab-based assay to such a miniaturized assay. Now, what was done here also in the NGM project was then using that cartridge, which can perform the assay, and equipping it with the microarray, which in our case here was spotted on a roll-to-roll spotting facility at Joanneum Research. And Basically, what you see here is some corner markers, the microarray in the middle, and this is sticking to the cartridge you see here on the left, and this is the array layout on the right. And then this cartridge runs in our reader, and on the left side you see again the comparison between symmetric and on the right side the late Psi PCR, symmetric Psi PCR on the left side, and the late one on the right side. And here you can basically exactly see what we've seen before. Very strong signals for the late PCR, well, for the symmetric. Very weak signals, even though we still can detect them. Of course, speaking of multiplexing, this can be also multiplexed. So here you see a duplex late Q Psi PCR. Sorry for the whole of that, but it's a combination of everything here. So it's a Q PCR, but this special method, and a late one. And this nicely works also for the two genes we are looking for SARS-CoV-2. And this also can be hybridized. So this was still the Q Psi PCR, but you can also run it as an endpoint Psi PCR and equip it with labeling, and then you can hybridize it and see something like that. And again, you see very nice spots here, which you easily can quantify. And this brings me already to the summary of what I showed you today. Very quick summary. I showed you today some aspects and strategies to consider for lab on chip, either lab on chip development or lab on chip fabrication, whatever. For molecular diagnostics, things to consider always are the level of integration, the level of multiplexing. I told you why high level of multiplexing may be important for the future, and also why high level of integration may be the future, and why speed plays an important role, and how this can be achieved both for amplification and also for hybridization. And then I showed you two examples, one on bacterial species and one on virus identification. And by that, I would like to thank you for your patience, and it's question time then.